Good afternoon. Welcome to our digital day lecture. Because classes were canceled because of snow today, I'm just record it as if I was teaching you all in front of class. Obviously, you guys can't answer any. I can't answer any of your questions. You guys can't ask questions. But if those do arise, feel free to bring them up next time we meet, or send me an email or whatever. Just some announcements and reminders. So today, or tomorrow, we will still have lab as regularly scheduled, as long as classes don't get canceled in. And we'll be starting an animal physiology lab where we will be looking at the diving response in humans. So come prepared to get your face dunked in water. If you have long hair, maybe pull it back in a ponytail. Don't wear lots of makeup. And bring your sense of adventure, because this is going to be fun. And then we are, as the title indicates, starting animals, which we'll talk about for the next few lectures. And this is going to be an introduction to basically characteristics about animals, which we use to help classify them into different groups. Next time we'll go into invertebrates and the phylogeny there. Okay, so first off, what is an animal? I think the best question to really help with this, or help kind of clear up some misunderstandings, is when I ask what is an animal, or if I ask is an insect an animal, some people will say no, because generally you think of, all right, birds and mammals and lizards and snakes and stuff like that. But animals are encompassing quite a bit of organisms. So anything that's not a plant and not a fungus that can move, that's crawling around, is an animal. The characteristics are a little bit hard to define, but there are some traits which animals all share. Okay, the first is, or one of them is, they are multicellular. And this isn't unique to animals. Fungi are multicellular, many of them. But all of them are multicellular. So we don't have any unicellular animals. And generally, actually, they have greater than a few cells and are visible to the naked eye, right? And being multicellular, they are also eukaryotes. They have an abundant protein called collagen. Collagen is unique to animals, and this is the most abundant animal protein. And they generally also contain tissues, although there is one group that does not have tissues, which is the peripherans, the sponges. They are also heterotroph, so just like fungi, they rely on other organisms for energy, or they have to eat other things in order to gain energy, and they cannot photosynthesize or get energy otherwise. They also have sexual reproduction in their life cycle. Some of them can also asexually reproduce, and we've talked about that when we talked about reproduction way back at the beginning of class. But generally, all of them can sexually reproduce, or have the potential to. And that is animals. Okay, so we're going to switch and talk about kind of the evolution of animals within our timeline, or at least the events in which evolution occurred. We'll then go back to some of the developments or the developmental stages in animals after this. So I wrote on this timeline some of the events which we have already studied. So our eons are the Haldean, Archean, Proterozoic, and Phanerozoic. And animals appear in the Proterozoic, in the late Proterozoic, which is also referred to as this last section here, or Neoproterozoic. I'll just draw an arrow here. So Neoproterozoic is where animals occur at about between 1 billion years ago. I'll just write a 1. And 542 million years ago. So this is an era which is a time period shorter than an eon. From 542 million years to 251 million years ago, we have the Paleozoic era. And then from 251 million years to 65 million years ago, we have the Mesozoic. And finally, the Cenozoic. So we'll talk about the major events within there. And we again, previously talked about how the Earth was formed, prokaryotes evolved, and then we have the evolution of photosynthesis through cyanobacteria, then eukaryotes, and we're going to kind of pick up where we left off there with our animals. So, in some time in this Neo-Paleozoic era, we have animals which evolved. So somewhere around here. And within that 500 million years, you have actually quite a bit of evolution occurring. This is a very long time period. We aren't drawing this timeline to scale. But it appears that the first animal-like organism which occurred was a sponge, and most animals at the late Neo-Paleozoic era were part of what's called this Edicarian biota. And these are mostly soft-bodied animals, which include things which we see today like jellyfish and sponges. But there wasn't a lot of form and structure to them. They were pretty simple and mostly soft-bodied. Okay, so this is at about 565 million years ago, we see the radiation of this Edicarian biota. And animals remained like that for quite some time. In the Paleozoic, however, we have one, I'm going to use a different color, sorry. We have a very important time period called the Cambrian era, where we had the Cambrian explosion. Okay, and during the Cambrian explosion, you have a diversity of body forms which occurred, and most invertebrate phyla show up in the fossil record during this Cambrian explosion. Including, there are also the transition from sea to land animals also occurred. All right, the Mesozoic is very important because you had a lot of diversity which occurred here. You start to see animals such as birds and mammals. And what's important to birds and mammals are flowering plants. So this gave a new source of food for mammals and birds to rely upon for their food, and allowed for a diversity of animals to arise from that period. The last one, the Cenozoic, included the mass extinctions of dinosaurs and the rise of large mammals. Okay, so we basically filled in our timeline from the beginning of time to current time period with the evolution of animals. And we talked about some of the plants, including flowering plants, in there as well. Okay, so now we're going to go back to some of the development of animals. So animals, as we know, sexually reproduce, and they do that through the fusion of sperm and egg. Now, generally, we've got in animals, eggs are produced by female and sperm by male. Those are the symbols for each. We'll use those throughout the course. Eggs generally are remain within the female and are larger and more energetically costly. Sperm is smaller, generally goes to the egg where fertilization then takes place. And when fertilization takes place, you have two haploid nuclei which fuse then into a diploid nucleus, and then a zygote is formed. Okay? But after the zygote forms, it then goes into multiple rounds of what's called cleavage. All right, what happens with cleavage is the cell splits, it goes through mitosis, but the overall mass of the now multicellular organism does not change. So the cells get smaller and smaller. After multiple rounds of cleavage, you then have a blastula forming. And a blastula is essentially a large sphere of these cells that have gone through many rounds of cleavage, right? So I'm showing basically a cross section, but if you looked at these, it just looked like a round ball. On the inside of your blastula, you have a blastocele, C-O-E-L. That suffix "cele" will come up again and again. It refers to a space, an open space. So it will be filled with, fluid or air, but nothing substantial in a cavity. Okay. So now that a blastula is formed, it'll continue to develop and then go through another form of development called gastrulation. Okay, gastrulation is the development of embryonic tissues. Okay, these are tissues that will then become different parts of the body. So what this looks like, though, is that hollow ball starts to invaginate, or part of these cells here at the bottom will push up and form this space, okay? And so then you have a gastrula forming. Now there are different parts of the gastrula, so we'll draw another one down here. Okay, now the space on the inside here is still the same place that it was in the blastula. So this is the blastocele. Okay, but now you have an inside and an outside layer forming. Okay, so this outside layer, which I'm going to draw as blue now, is the ectoderm. Okay, the ectoderm is going to be, or develop into the outer layer of the organism. So, for example, in humans, this is going to form our skin. It forms other things as well, so our nervous tissue forms from there as well. But the outer layer generally is what forms from the ectoderm. Okay, and then it has an inner layer forming as well. I'm going to draw that in yellow, and that's our endoderm. Okay, this is going to form the inner layer in organisms. So this is going to form in humans the digestive tract. Okay, your inner tube forms from that. Now, all animals have these two embryonic layers, ectoderm and endoderm. In some, however, it's also going to form a third layer. Okay, and I'm going to draw that in red here, and we'll talk more about this layer as well. But this then would be the mesoderm, which would form internal, we're going to call it the middle layer, right? Would form internal organs and muscles and things between the skin and the digestive system, essentially the digestive tract. And again, we'll talk more about these in a minute. All right, so that then is the process of gastrulation, or the process of making then embryonic tissues, which will eventually form the different parts of the body of the animal. Okay, so we're going to move. Okay, there are a couple other important structures I forgot to talk about. So in purple, so this little hole then here at the bottom, that's purple. It's not there, I'm just drawing it, but that then is called the blastopore. And this looks ugly because I'm using my finger because my pencil's dead. But blastopore. Okay, and then one other layer, or one other structure. This inside layer here then is called the archenteron. Okay, so those are then the different parts of our gastrula, since there's no questions. All right, so again, we talked about the blastocele before, which is an inner space in the blastula. But as the body forms, so this is this is not the small cellular level, which we're talking about with the blastopore, the blast. Oh, there are a couple things I forgot. Hold on. Okay, so the body cavities then, sorry about that before, are areas within the body. So this is looking more like, a worm or an adult organism that's fully developed and functional within its environment. It's going to have spaces within its body, and that's what the body cavities are referring to. Again, we see this cele, C-O-E-L, in here, and that's referring then to that body cavity. An acoelomate does not have a body cavity. Okay, so if we have its ectoderm here, its mesoderm inside of that, and its endoderm forming its digestive tract. Okay, and this is going to be completely filled in. There's no space, so there's no coelom, right? So that A in front of it means not, or generally. So acoelomate, there is no coelom in acoelomates, okay? All right, a pseudocoelomate. Okay, this is the ectoderm. It has an inner endoderm. And then another layer of the mesoderm. The mesoderm touches the ectoderm, but does not touch the endoderm. So then that area is the pseudocoelom. Pseudo meaning false. Okay, so that area right there in between the ectoderm and the mesoderm is just space. And that's the pseudocoelom. All right, coelomates then have a slightly different body cavity. So we still have our ectoderm, and we still have our endoderm. But the mesoderm goes around the ectoderm and around the endoderm and into the out to the ectoderm. So this is all filled in here. See that? So basically the coelom forms with their coelom, that space then is formed by completely the inner mesoderm layer, right? Whereas the pseudocoelom is formed by the mesoderm and on the outside and the endoderm on the inside. Okay, so another way in which we can classify different animals is in their development. And two forms of development have been classified which show specific characters: protostome and deuterostome development. And I'm going to draw and kind of list out these different ways that they develop. So first, I'll just go through deuterostomes, and then we'll talk about protostomes. So first off, in the cleavage. So if you remember, we talked about cleavage is cell division without increasing in size. So the cells, or the overall size, so the cells themselves actually get smaller. In deuterostomes, what happens is when they split on top of each other, they actually remain in the same axis. Okay, so this kind of line here where they split is the same as you're going through multiple different levels, okay? And this is called radial cleavage. It's radial. Okay, now if one of these cells is split off from the rest, in deuterostomes, this one cell can still go through cleavage and become a viable embryo. Okay, and this is called indeterminate development or cleavage. Okay, so if you can split off all these different cells early enough in their development, you could basically create clones. This is kind of how identical twins occur in humans. Okay. All right. Later on in development, when you have the gastrulation occurring, right? And if you remember, this hole at the bottom is called the blastopore. Well, eventually, this invagination is going to go all the way through and form a tube. That tube is going to be, remember that inner lining is going to be the digestive tract. Well, once it becomes then, let's say this becomes a full worm. Okay, and then that tube running through it okay, there's one end, and I'm just going to write little back brackets there. There's the other end. The blastopore then becomes, in deuterostomes, the anus. Okay, so the blastopore eventually becomes that. And then the other end, where it goes all the way through, that becomes the mouth. Okay, there's his little eye on this worm. Okay, and then the last kind of similar developmental thing, when you have your gastrula, the mesoderm forms from the coelom forms from these out pockets of mesoderm. Okay, and then that will eventually become, those out pockets will eventually become the coelom. Right, and you got this is the digestive tract. Anyway, something like this. All right, I'm going to color in the coelom black. So this on this side, this on this side. Okay, that's deuterostomes. Okay, protostomes, let's go back here. They don't have radial cleavage. Instead, what happens is the that line in which the axis forms rotates half of a cell, and this is called then spiral cleavage. All right, so this division between two cells then becomes the center of the next cell. Okay. If one of these breaks off, so now you have this sort of arrangement, it actually will not become an embryo. Okay, so if one of the cells comes off, that messes everything up, and development doesn't occur. So this is called determinate cleavage or development. All right, so pretty much everything is the opposite as the other one. So we have then our gastrula, our blastopore. In protostomes, you've got your little worm here. The blastopore becomes actually the mouth. And the other end up here then becomes the anus. So the blastopore becomes the mouth in protostomes. And the coelom, rather than forming from an out pocket, forms from a split in the mesoderm. But in the end, it forms the same coelom-like structure. All right, so that's the difference between protostomes and deuterostomes, or 4 major differences between the two. And we'll talk about when we go through our invertebrate phyla next time, we'll talk about these different deuterostome and protostome development. We'll also talk about symmetry, so radial symmetry, and these other things which we've talked about in lab when we classify different animals. So radial symmetry, pentaradial symmetry, bilateral symmetry. And all of this will then apply to our different groups. And now you have the background to understand all those things. Okay, and that's it. That's for the lecture. There is a nice lengthy worksheet for you to do on MySVU. And sorry, we can't do Kahoot. Maybe we can do that on Monday.